Hello, welcome to this Emmy Foggy Dog event. I'm Sally Callow. I am the founder and manager of Emmy Foggy Dog, a UK social enterprise that works for the benefit of the Emmy community. I also founded Stripey Lightbulb CIC. We offer online training to professionals about what Emmy CFS is and is not. I'm also an Emmy campaigner and have been since 2014, but most importantly, I'm a person living with Emmy since 2006. This session has come about because of the continuance of misinformation about what ME is and is not. So I want to talk about it through the lens of biomedical research. So welcome to this session. As you can see, the title of the session is called Recent Advances in Biomedical ME Research and Why This Field Needs More. Now, this is going to be presented from a patient-informed analytical perspective, and the main aims of the session are to highlight examples of strong biomedical research from the past year to make sure they're relevant and completely up to date, to identify what these studies do well, and also to outline why this field warrants serious research attention. A little bit of context first. So that's National Institute for Health and Care Excellence. The NICE committee acknowledged in their 2021 guidance that people living with ME/CFS frequently research their condition extensively, often becoming well-informed and sometimes more knowledgeable than generalist healthcare professionals about the available evidence. Patients may scrutinise treatments and research claims closely, especially where previous interventions were perceived as harmful or not evidence-based. I encourage anyone to research the PACE trial, just as a case in point. This contributes to a culture of critical engagement with research and medical advice, rather than passive acceptance. The evidence review also shows why some patients developed increased self-reliance and independence from medical advice when they felt professionals misunderstood the condition or relied on contested models. So basically, the community keeps up to date with the latest research. With that in mind, I would encourage any early career researchers or researchers in this field to sign up to Science for ME. This is the Science for ME website. Please do sign up and use this really valuable resource. Science for ME describes themselves as an independent, patient-led international forum for people with ME/CFS and their carers, clinicians and scientists and advocates who support them. It's a great opportunity for researchers to discuss ideas and research with the community. As an advocate, I can't tell you the amount of times since 2014, Science for ME have stepped in and advised scientists on a way of making their research stronger or flagged problems with research studies. Their knowledge and expertise are invaluable, so I'd thoroughly recommend anybody signing up. If you've got an interest in ME/CFS biomedical research, signing up to this website and forum. I will include the link to Science for ME in the resource document that will be attached to this event. CFS research has been massively underfunded for decades, and this causes immense difficulties and barriers to effective treatments and potentially cures and biomarkers. If this was an advocacy webinar, this subject alone could fill a webinar and more, so that's for another day. This section is about learning from what works, not criticising individual researchers. So across complex diseases, methodological challenges are common, but in ME, their impact has been particularly significant. Studies using broad or inconsistent case definitions have often grouped together patients with very different underlying conditions. This can dilute biological signals and make findings difficult to replicate. Similarly, where post-exertional malaise hasn't been properly accounted for, key aspects of the disease process may be missed entirely. particularly in studies that only assess patients at rest. There has also been a tendency in some areas of the literature to rely heavily on subjective outcome measures without accompanying objective data. That can make results harder to interpret in biological terms. In addition, small sample sizes or poorly matched control groups can limit the strength of conclusions, even when research question itself is well framed. And finally, early or limited findings have sometimes been overinterpreted, which can lead to cycles of enthusiasm followed by uncertainty. Individually, these are familiar challenges in many different fields, but taken together in ME, they have often slowed progress by obscuring underlying biology rather than clarifying it. In terms of methodological implications, strong research in ME have these things in common: heterogeneity of ME populations, importance of clear case definitions, the inclusion of post-exertional malaise always. To be clear, if you do not include post-exertional malaise within your biomedical research study, are you really researching ME? Because that's a defining characteristic of this disease, and it impacts so many different processes and systems. Appropriate control groups and avoiding overinterpretations. So how weaker studies have historically slowed progress in the field. Researchers have used broad or inconsistent case definitions, failure to account for post-exertional malaise, over-reliance on subjective outcomes, underpowered or poorly controlled studies, over-interpretation of limited findings. So for the purpose of this webinar, I've chosen 3 examples of biomedical research published internationally in the last year that demonstrate methodological care and biological relevance. I'm going to talk about each one individually, what the researchers did, what they found, and what the possible next steps could be. And then I'm going to talk about the combined strengths of these 3 studies when you take them collectively. So this is the first research study that I chose. It was published in April 2026, so this year, and it was by Braji et al. And what the researchers did was they looked at the fluid around the brain and spinal cord, so that's cerebrospinal fluid or CSF, in people with ME/CFS. They studied 31 patients with ME/CFS, measured over 900 different proteins in this fluid. They then compared these protein patterns with how severe the illness was, whether the patients had postural orthostatic tachycardia syndrome, POTS, for those of you that don't know, which is a common condition in ME involving dizziness and heart rate issues. So in simple terms, they were trying to see if there are biological fingerprints in the brain that match different ME symptoms. So what they found was there are clear biological differences inside the nervous system. They found distinct protein patterns in the brain and spinal fluid of people with ME. These patterns relate to immune activity, inflammation, and metabolism. This means that ME is showing measurable biological changes, not just symptoms. In terms of links to symptom severity, certain biological pathways were more active in people with more severe ME, including immune system pathways, blood clotting processes, and inflammatory responses. And this means the sicker someone is, the more these biological changes show up. In terms of links to specific symptoms like POTS, people with POTS and ME had signs of immune cell activation and platelet or clotting activity. This means different symptoms in ME may come from different underlying biological processes. This is exciting because in terms of possible early biomarkers, the study identified specific protein ratios that track with how severe someone's illness is. So in theory, these could eventually lead to diagnostic tests or ways to measure how ill someone is. For this particular research study, the overall takeaway is that ME/CFS has real measurable changes in the brain and nervous system, and different symptoms and severity levels may be caused by different biological pathways. Why is this considered good research? It studies the brain directly, which is rare in ME research. Most ME studies use blood. And this particular research study uses cerebrospinal fluid, which is much closer to the brain and central nervous system. And this is important because ME likely involves brain immune interactions. This study also uses advanced high-quality technology, measures 900 proteins using proteomics, so mass spectrometry. This allows very detailed, objective biological data and much deeper insight than basic tests. It also links biology to real symptoms. So instead of just finding abnormalities, it connects them to severity and specific conditions such as POTS. And this makes the findings clinically meaningful and not just theoretical. It also supports the idea that ME is not one single disease. The different protein patterns equal different subtypes of ME, which is really exciting to see biomedical evidence of this. And this helps explain why patients experience very different symptoms, and that one-size-fits-all treatments do not work. They might work for one particular subset, but the vast majority of ME patients find it doesn't work for them. And this research study also does transparent and reproducible data. The data is shared in a public repository, so Proteomics Change. Again, a link will be put in the resource at the end of this session. And this is a hallmark of good, trustworthy science and allows other researchers to verify and build on findings. And lastly, this generates testable future directions. Where can we go from here with this research study? It identifies specific pathways: immune activation, coagulation, so clotting, and neuroimmune signaling. These are clear targets for future studies and drug development. But if I'm going to give a balanced view, although this is strong research, it has its limits. It's a small sample, only 31 patients, and needs replication. And there is no healthy control group in this data set, so that limits comparisons. And it's mostly exploratory, finding patterns but not proving a cause. So it's very promising but not definitive yet. So the bottom line of this first research study is that this is good, high-quality biomedical research because it uses objective biological measurements, focuses on the brain, links biology to symptoms, and points towards biomarkers and subtypes. And most importantly, it adds to growing evidence that ME/CFS is a complex biological disease with measurable changes in the nervous and immune systems. This study was published in August 2025 and was by Moise et al. This study looked at a blood protein called haptoglobin in people with ME/CFS. The research included 140 ME patients, 44 healthy controls, and the patients were tested before and after a controlled physical stress test to trigger post-exertional malaise. They analysed the levels of haptoglobin, the different types or genetic forms of haptoglobin, and the cognitive performance. What researchers found was that there was a biological change after exertion. ME patients showed a drop in haptoglobin levels after exertion. This means that the body responds abnormally to activity. So this backs up what patients experience with PEM and have been saying anecdotally for decades. It also links to cognitive problems, so brain fog. Lower haptoglobin levels were linked to worse cognitive performance. This means this protein specifically may be connected to the brain symptoms with ME/CFS. Different genetic types affect severity. So certain haptoglobin types matter. HB2-1 type is more common in ME and linked to worse post-exertional malaise and worse cognition. HP1-1 type is linked to milder symptoms and better cognitive function. And this means people may experience different forms of ME depending on biology or genetics. This research study also shows structural changes in the protein itself. Researchers found physical differences in the protein structure in ME patients. And this means this isn't just about levels, the protein behaves differently in the body. As a potential biomarker, haptoglobin could be used to measure post-exertional malaise severity or help identify subtypes of ME. So the overall takeaway is that this study shows that ME patients have measurable biological changes after exertion. A specific blood protein is linked to post-exertional malaise, brain symptoms, and disease severity. And it supports the idea that ME has real biological causes and different subtypes. So why is this good research? One, it's a large sample size, large by ME terms, because our sample size is usually quite small, with 140 patients. That's relatively big in this field, and it's much stronger than small pilot studies. It includes a control group, compared ME patients to matched healthy controls, and this is essential to show changes are disease specific. And it uses a real physiological trigger, so post-exertional malaise. Instead of just measuring it at rest, they triggered symptoms using exertion, and they measured biological changes before and after. And this is a major strength because post-exertional malaise is the core symptom of ME. And many studies fail to capture it. This was a longitudinal design, so before and after, and changes were measured within the same patients over time. And this is stronger than a single snapshot and shows dynamic disease processes. There's an independent validation cohort, so findings were tested again in a separate group of patients, and replication is a key marker of good science. This study combines multiple types of analysis, so proteomics, protein measurement, genetics, so protein types, and functional testing, cognition and exercise response. This multi-layer approach is much more powerful than single method studies. And this study in particular has direct clinical relevance. It links biology to post-exertional malaise, which is the core symptom of ME, and cognitive dysfunction. And this makes it useful for diagnosis, for stratifying patients and potential treatments. But to give a balanced view, this is really good research, but it's still not definitive. Association does not prove cause. Findings need to be replicated in more diverse populations, and post-exertional malaise induction in a lab may not fully reflect real-life severity. So the bottom line is this is strong, high-quality ME research because it uses a large, well-designed cohort, measures the real biological changes during post-exertional malaise, identifies a specific biomarker linked to symptoms and severity, and supports the idea that ME has distinct biological subtypes. But most importantly, it provides some of the clearest evidence yet that post-exertional malaise has a measurable biological basis, and it is not just a subjective symptom. So this is the third research study. This was published in December 2025 by Germain et al. And what the researchers did was they looked at what happens in the blood after exercise in people with ME/CFS, specifically focusing on post-exertional malaise. This study included 79 people with ME/CFS, 53 healthy but sedentary controls. It measured 6,000 proteins in the blood, so very large-scale analysis. Participants did 2 exercise tests 24 hours apart to trigger post-exertional malaise, and researchers tracked changes before exercise, after exercise, and during recovery. In simple terms, researchers were asking what goes wrong in the body after activity in ME patients. What they found was major biological changes after exercise, not just at rest. The biggest differences appeared during the recovery phase when PEM happens. So this means ME isn't just a baseline illness. The problem shows up most clearly after exertion. It showed ongoing maladaptive response. Instead of recovering normally, ME patients showed prolonged disruption in their immune system, metabolism, and muscle neuromuscular systems. This means the body gets stuck in an abnormal response after activity. Immune system dysfunction. So key immune pathways were suppressed or altered, including T cells and B cells and IL-17 signaling, which is an important immune pathway. This means the immune system doesn't respond or reset properly after exertion. In terms of energy metabolic problems, the evidence of mitochondrial stress and increased reliance on inefficient energy pathways means the body struggles to produce energy normally after exertion. So these protein changes were found to be linked to real symptoms, such as symptom severity, exercise capacity, and symptoms like pain, sore throat, and fatigue. These biological changes directly relate to how ill someone feels. This study also showed differences between men and women. Males and females showed different biological responses. ME may behave differently biologically in different people. So this study shows the biggest problem in ME happens after exertion, not just at rest. The body fails to recover properly, and this affects the immune system, energy production, muscle function, and these changes line up directly with post-exertional malaise symptoms. So why is this good research? Well, for one, it studies the core symptom of the disease. PEM is a defining feature of ME, but many studies ignore it. This study deliberately triggers post-exertional malaise on purpose, measures the biological response, and that is a huge strength of this study. It directly studies what matters most clinically. It's a longitudinal study, so it measures before, during, and after exertion, not just a single snapshot, and it shows cause and response patterns, not just differences. It included a large and well-controlled cohort, so 132 participants in total, and included matched sedentary controls. It's a massive biological data set. It measured thousands of proteins, so more than 6,000, and that's very advanced and provides a deep systems level view. This study also links biology to symptoms and performance. It connects molecular changes to symptom severity and exercise capacity, and that makes findings clinically meaningful, not abstract. In terms of its methodological design, it's strong. It uses controlled exercise challenge, so CPET, multiple time points, and advanced proteomic technology. This is considered high quality experimental design. It also addresses a key criticism in ME research. A common criticism is patients are just unfit or deconditioned. This study countered that by using sedentary controls and showing distinct biological responses. This is incredibly important scientifically and clinically. So to give a balanced view, this study does have limitations. It's still exploratory, so it identifies patterns but not exact causes, and the complex data needs replication. And the exercise testing may not reflect all severity levels, particularly very severe patients who are most often bedbound. But it's a very good research study. I would encourage anybody that's watching this to read the study for yourself. The bottom line is this is one of the strongest ME studies because it directly examines post-exertional malaise, shows clear biological dysfunction after activity, links immune, metabolic, and muscle systems together. If ME-CFS is a disease where the body fails to recover properly after exertion, And this study shows that clearly at a biological level. Now let's talk about the big picture. These three studies all focus on biology in ME/CFS, but from slightly different angles. The first study, CSF Proteomics, studied the brain and nervous system biology. The second one, the haptoglobin study, was a specific blood biomarker and post-exertional malaise study. And exercise proteomics was studied in the third one, whole body response to post-exertional malaise exertion. Together, they answer what's happening in the brain, blood, and body, especially after exertion. So when you compare them all, the takeaways are ME/CFS is a brain-body disease involving immune and metabolic disruption. ME is fundamentally a disease where the body fails to recover after exertion. ME involves abnormal immune regulation, especially after stress. The body struggles to produce and regulate energy properly. There is no single ME type, so there are multiple biological subgroups that exist. And altogether can be summed up as ME/CFS is a condition where immune and energy dysfunction affects the brain and body, especially after exertion, and varies between biological subtypes. As you can see on the screen, these studies collectively provide protein signature, a candidate marker, and timing insight. And this is what is needed before building a diagnostic test. In terms of what happens next, we need larger validation studies to test whether findings replicate in different countries and different patient groups. We need to combine markers. So not one biomarker, but a panel of biomarkers. And we need to standardize testing conditions. So we likely need challenge tests, so before and after exertion, and not just resting blood tests. We need to stratify patients, so precision medicine. These studies show different biological patterns. So the next step should be to divide ME into subtypes based on biology, not just symptoms. This will improve the chances of diagnosis, improve clinical trials, and enable targeted treatments. We also need mechanism-focused research, and key areas to investigate further are immune dysregulation, so T cells and IL-17, mitochondrial energy pathways, oxidative stress, so haptoglobin findings, and brain immune signaling. And the goal from that is to move from association to cause and mechanism. We can also look toward treatment development. Once pathways are confirmed, research can move to immune modulating therapies, metabolic treatments, oxidative stress interventions, and personalized interventions based on subtypes. There has already been some integration with long COVID research, but they need to be stronger. And these findings strongly overlap with long COVID. So post-infectious onset, post-exertional malaise biology, and immune metabolic dysfunction. So the next step with that is to use larger long COVID cohorts to validate biomarkers faster and accelerate treatment discovery. So one key message that I want you to take away from this section is these studies don't just show that ME/CFS is biological. They show exactly where and when to look for measurable biomarkers, bringing the field significantly closer to a real diagnostic test. What do these studies tell us about the state of the ME/CFS research field in 2026? These recent studies all point to the same conclusion. ME/CFS is clearly now being recognised as a real biological illness, and researchers are starting to understand how it works. What the studies tell us: one, it's not all in your head. Scientists can now measure real changes in the brain, blood, and immune system. Two, the biggest problem happens after activity, so post-exertional malaise means the body doesn't recover properly from exertion. This is now measurable in biology. Fantastic. The immune system and energy systems are not working properly. This helps explain symptoms like fatigue, crashes, and brain fog. It's not one single illness. There are likely different subsets of ME with different biology. And what this means for the field is that the research field has moved significantly forward. Before, it was, we don't really know what ME is. But now we can see where the problems are and start measuring them. ME research is shifting from proving the illness exists, which is something that I found incredibly frustrating as a campaigner for the past 12 years, to identifying biomarkers and developing tests and treatments. Good researchers, we need you. If you're looking for a problem that is still genuinely open, ME/CFS is one of the more intellectually demanding areas in biomedical research right now. It's a multi-system illness with signals across immunology, metabolism, and neuroscience, which makes it a methodologically interesting space if you like working across data sets, integrating approaches, and dealing with imperfect but very real-world biology. There's also a clear clinical gap. This is work that is directly clinically meaningful. No validated biomarkers, no definitive diagnostics, and very limited treatment options. That gives you a rare opportunity to work in a space where foundational ideas can shift the field. The field is also very open to new collaborators. A lot of progress will come from people bringing in fresh biomedical perspectives, whether that's from adjacent disease areas, systems biology, or newer computational approaches. You're all welcome. Early career researchers are particularly valuable here. There's space to contribute quickly, shape questions, and do work that's recognised as needed, not incremental. One thing worth being aware of and leaning into is the role of patient knowledge. As I've already mentioned at the start of this talk, this is a field where respectful engagement with people living with the condition really matters. We are the experts on this condition until researchers actually find out the biological nature of our disease. Please engage with us, not as a token exercise, but because it often improves study design and helps keep the science grounded. If you're comfortable working in a complex, evolving space and want your work to matter beyond publication, it's a field that's worth your attention. The ME/CFS research field has clearly shifted. We're no longer asking if this is a biological condition. The evidence is there. What we're now starting to understand is where the dysfunction sits, when it shows up, especially after exertion, and what we might be able to measure. That moves the field much closer to biomarkers and meaningful clinical progress. It's still a complex space, heterogeneous, multi-system, and not fully mapped, which is exactly why it remains scientifically challenging and worth working on. There is a real need for new collaborators, fresh biomedical perspectives, and interdisciplinary approaches. And more importantly, research here works best when it stays grounded, engaging respectfully with patient knowledge to improve the science itself. This is an open field, but one that now has enough biological signal to build on. Please come and join us. Thank you.